In this video, I am going to review about the chemical structure of nucleotides. There will be 4 critical points we will be learning here. 1. What is nucleotides and why it is important? 2. What are the important chemical structures that build nucleotides? 3. What are the differences between DNA, RNA, and ATP, as the 3 of them are nucleotides? 4. How do complementary base pairs form double helix in DNA? Okay, let's go! A nucleotide is the basic building block of nucleic acids. RNA and DNA are polymers made of long chains of nucleotides. A nucleotide consists of a sugar molecule, either ribose in RNA or deoxyribose in DNA. This sugar molecule is attached to a phosphate group and a nitrogen-containing base. Both DNA and RNA are built with a sugar backbone, but whereas the sugar in DNA is called deoxyribose, the sugar in RNA is called simply ribose. The deoxy prefix denotes that whilst RNA has 2 hydroxyl groups attached to its carbon backbone, the DNA has only 1 and has a lone hydrogen atom attached instead. RNA's extra hydroxyl groups are useful in the process of converting genetic code into mRNAs that can be made into proteins, whilst the deoxyribose sugars give DNA more stability. Nucleic acids are composed of combination of 5 nitrogenous bases: guanine, adenine. Those 2 are double-ringed purine molecules, while cytosine, thymine, uracil are single-ringed pyrimidine molecules. Thymine and uracil are chemically similar molecules. Thymine is present in DNA, while uracil is used in RNA. Okay, next. Nucleotide is also an important structure in the energy metabolism, as it is the basic structure of chemical energy adenosine triphosphate, that often abbreviated as ATP. ATP is the common goal of many catabolism that correlated to the energy production mechanism. In Krebs cycle, guanosine triphosphate or GTP is produced during succinyl conversion into succinyl-CoA. However, in some reference, this reaction mention ATP as the product instead of GTP. This is because they are equal in energy level, and due to the similar purine ring structure, GTP is easily converted into ATP. Okay, next. Now let's talk about DNA. DNA has a double helix structure with sugar and phosphate on the outside of the helix, forming the sugar-phosphate backbone of the DNA. The nitrogenous bases are stacked in the interior in pairs, like the steps of a staircase. The pairs are bound to each other by hydrogen bonds. The pattern of complementary base pairing are adenine will always form 2 hydrogen bonds with thymine or uracil, while guanine will always form 3 hydrogen bonds with cytosine. The 2 strands of the helix run in opposite direction, so that the carbon number 5 end of one strand will face the carbon number 3 end of its matching strand. This anti-parallel orientation is important for DNA replication and also in many nucleic acid interaction. Okay, that's a wrap. So this is our review points for nucleotides. After watching this video, can you answer these questions? 1. What is nucleotide and why it is important? 2. What are the important chemical structures that build nucleotides? 3. What are the differences between DNA, RNA, and ATP, and how do nucleotides form them? 4. How do complementary base pairs form double helix in DNA? Okay, that's all for today's review about nucleotides. For questions and comments, you know where to contact me. See you on another lecture. Bye!